Hello all. So today we're gonna have a little bit different. Since I'm out of town, I did want to. We just don't have a lot of time in class, so I wanted to make sure that everyone was able to um, get the full information about genetics. We are starting in the genetics section. I know it can be difficult for some people. So this recorded lecture, it's something that's called a flipped classroom approach. Basically, you're going to listen to this lecture, and then on Wednesday, we'll come back into lab and we will work through some of the more complex concepts. If you don't listen to the whole lecture, though, you will be behind for the lab section because there is a activity that you're going to do on your own and then come present to the class. So similar to our discussion section in lecture, except it's going to happen in lab. And you do need to make sure that you are prepped for just understanding the basic concepts, because if you don't, then again, you're going to be a bit behind and it'll be hard for you to keep track. All right, so first, I know that there's no early or late to this lecture, but this is your bonus slide. Again, you can go back and look at this. But this is your bonus slide, and basically, this is one that could be on the final. As long as you are listening to this recorded lecture, you will be able to get the points for that. All right, so first we're going to finish up lecture 6. We talked about polycultures, companion planting, etc. So we're just going to run through that, and then we're going to jump into the genetics. All right, so we talked about niches in the last lecture, and the goal for polycultures, again, is to, let's say you want to grow multiple species. We talked about the importance of growing multiple species in a agroecosystem to make sure that it's a resilient ecosystem. So for polycultures, you want to choose multiple niches. So you don't just want to, if you want to grow multiple species together, you need to make sure that the niches are not overlapping, because we saw before that the overlapping niches would cause competition. So you need to think about soil type and pH, sunlight, growing zone, what the different plants need. Are they adding to the soil? Nitrogen fixation, soil building. Also, how do humans use them? Food, fiber, etc. And think about those pieces. So this is just a concept of a polyculture. And so you have the blueberries, lingonberries, and bunchberries. They're different sizes. They use different. They have different roots. They use different areas in the soil. The bunchberry that is at the base, like shade. The blueberries like full light, and lingonberries are in between. So you can see that you can grow them all together. And so this picture is a picture of sunflowers, which love sun, obviously, and they are growing at the base of that. There's kale and lettuce, and those need a little bit less light and are happy being shaded by the sunflowers. It's also another concept known as companion planting. So this means that certain plants perform certain functions in the soil, and because they're adding things to the soil or taking things away from the soil, a heavy nitrogen feeder won't like other heavy nitrogen feeders, that there are certain plants that work well together. So you can see that if you're planting beans on the top there, then planting beans with corn or cucumbers or other things is good because these are heavy nitrogen feeders, and then the beans add nitrogen because they work with the bacteria, the rhizobium bacteria. So this is just a concept of another way to reduce competition is to use things that work with the soil differently, and there are bad companions too. So the onions being a bad companion to the beans because they put out certain things into the soil. All right, so we're going to be talking also about other species interactions, specifically herbivory and pathogens. Herbivory, we talked a little bit about deer and deer exploding in the population section, but we're going to talk about insect herbivory mainly, and the pathogens. So remember we talked about pathogens in lab and how you test for pathogen resistance. But first we're going to start out by talking about, remember we talked about population dynamics. This is a slide from the first lecture, and this slide says, it's talking about this thing called the pesticide treadmill. So there's an issue with broad spectrum pesticides is that if you apply pesticides and you kill everything off, you're going to leave a couple of vulnerable ones that maybe didn't get hit by the pesticide, and then you're going to have a couple of resistant varieties, things with a genetic resistance to the pesticide, and those genetic resistance one will then pass on those genes to a bunch more. You spray again, and now you have more resistant bugs, and then you have to use stronger pesticides or new pesticides, and it can be exhausting and bad for the environment. Future generations can become more and more resistant. With population dynamics and pesticides, remember I showed this slide briefly during the last lecture, but the idea is that, so on the top graph here, population size, if you just kill everything off, predator and prey. Remember we talked about the R versus K species. And the R species come back very quickly because they make tons of babies. But the K species, like the predators, they come back slowly. And so if you apply a pesticide that kills all of the predators as well, the pests come back super quickly, and now there's no predators to kill them. So it's kind of like if you killed off all the aphids and the ladybugs, then the ladybugs will come back very slowly because they don't create that many babies, while the aphid would be a boom. So actually you've made your pest problem worse. Even if you kill off all the prey, if the predator is dependent on only that prey to eat, then their population will decrease as well. And then the prey will again come back faster. It's a little bit less of a dramatic thing, so that happens on the bottom graph there. So there's the same issue with fungicides if you're trying to kill off a pathogen. And again, if you have a mostly vulnerable population to fungicide, and you have a couple of resistant varieties, and you kill off most of them, then suddenly the genetically resistant one will breed with everything. So this is the concept of natural selection. This is artificial selection because it's with fungicide, but it's the same idea. If there was something that killed off 90% of the population, that 10% had a resistant gene, then they're going to pass their genes on. So to combat this in agroecosystems, there's something called integrated pest management. It's where you work within population and predator-prey relationships of pest organisms to reduce their impact on crops. The idea here is that you're working with the cycles themselves. You're not actually trying to just kill off everything because, again, the pesticides cause more pest issues and it's not a sustainable system. So the techniques in integrated pest management are physically removing the pests, spraying with water, deterring with some sort of smelly thing instead of killing them off, and encouraging predators. So planting things where predators can live, and encouraging spiders and maybe even releasing ladybugs. Introducing genetic resistance to crops is another way, and we'll talk about that later. And then also this targeted and not broad-range pesticide use. So only using pesticides to kill specific populations of pests instead of killing off everything at once. So it's just a little pyramid to explain. So think of this sort of like the food pyramid. The things you're supposed to eat the most of are at the bottom. Well, the things you're supposed to do the most of are at the bottom of this integrated pest management. So first of all, doing things like sanitation and moisture management, reducing, remember we talked about this in lab when Dr. Luer talked about the strawberries where you're working on keeping certain pests out, you have to manage the moisture. And that's true for insect pests as well. The next step up is this physical and mechanical removal. Traps and something called exodust, which is a type of dust that basically it mechanically kills the bugs. It does not actually kill them by poisoning them. So you can't actually have a genetic resistance to it. And so you use a bit fewer of that. And then predators and parasites, releasing them or encouraging them. That's the biological control. And then the last one is the chemical control. Again, using very small amounts of chemicals, but very targeted. So larvicides, baits, repellents, things that remove them from the area, that prevent them from being able to reproduce, but don't kill off the entire population. So you can see that this is sort of a different way of thinking about managing pests, and that has to do with population. You can also encourage predators with hedgerow buffers. So planting flowering perennials to provide a place for predatory bugs like spiders and parasitic wasps. And pollinators can also overwinter. We're going to talk about pollinators in a bit. Another way is to also have this, and we saw in the previous slide that you had these hedgerows, these bushes near the field. And what it does is it makes this dilute gene pool. If you have a bunch of vulnerable populations that are not being hit because you're only spraying a targeted area on your field and you're not spraying any of the bushes, then when the resistant pests come, they breed with they breed both with each other and with the ones that are in the refuge, which dilutes their gene pool. They're breeding with non-resistant ones, which means that they're not going to pass on the resistance gene as much. It dilutes that. And so you can have it where you've mostly gotten rid of all the ones in your field and you have a very vulnerable population in the refugia, but you don't have the hugely resistant population in your field itself. Another way is this managing soils for pathogen reduction. We talked about the ways to prevent certain funguses or pathogens or diseases from hitting the strawberry crop, and one of them was just solarizing the whole field, putting black plastic over top and killing it all. Again, we want the soil to be alive, but if you have a really bad nematode or pest problem, you can kill everything in the soil and then start over. Another way is this last one, the managing the rotation of crops, and this was very important. Kim Lewers discussed how at Bark they have a very specific rotation that they do to make sure that the place that they start with is low in pathogens and overall pathogen free. They don't fumigate everything. They use the rotation of crops as a sort of natural fumigant. All right, now we're going to start on lecture 7. So we're going to be focusing on chapter 14 and 15. But the deal is we're not going to be following the book as much for this one. The book goes over genetic populations a little bit in 14 and 15, but doesn't go over basic genetics. We are going over basic genetics because this is an intro class, and the agroecology book assumes that you've already taken a basic genetics course, which I know a lot of you haven't. So I want to make sure that we know the basics of genetics so you can understand how it works to breed and pass on genes from one generation to the next. So we talked about mutualisms before. So we talked about inhabitational mutualisms. The rhizobium and the mycorrhizal fungi that live in the roots. That's an example of an inhabitational mutualism. Both species are benefiting and one lives inside the other. The other one is an exhabitational mutualism, means it lives outside. The organisms involved are independent physically, but they interact directly. And so these are things like pollinators. Ants and aphids have another thing where ants will actually farm aphids. They'll protect the aphids from harm, and the aphids in turn give them a sweet liquid. And but the pollinators are what they're focusing on in this intro into genetics. And then finally, there's this indirect mutualism. The interactions among a set of species modify the environment in which they all live. So this would be something like a nitrogen-fixing crop, or the cover crops that change the soil in some way that's beneficial to everything else. They're not trying to be beneficial to another species. It just sort of happens. That's why it's indirect. So mutualisms are important to have in an agroecosystem because they're key to establishing these sustainable systems that require fewer external inputs and less human intervention. So we're going to focus on pollination. So I'm sure you learned about basic pollination, but we're going to talk about the actual mechanism of it and how it happens. So pollination is the transfer of pollen from the male anther to the female stigma in a plant flower. Okay, so the anther is the orange one right here, and the stigma is over here. It is the female part. Okay, so we have the sperm going into the egg, and very similar concept here. And now, why is pollination important? Is important to understand. It's because it's sexual reproduction. That's very important for evolution. Sexual reproduction produces variable offspring. You want variable offspring because if you have the same offspring, monocultures and genetically identical plants like the banana, that's bad. If you have, you want to create diversity and variation among population, you shuffle the genes around, and now you have, some bananas in the population that are resistant to a disease instead of everything being the same resistances. You don't want to have constant asexual reproduction. And you need variation for natural selection to occur. Remember, natural selection is a good thing in terms of the resilience of plants and ecosystems. You want the actual natural selection to happen. All the plants that are susceptible to a disease or other things dying off, and having the stronger plants that are resistant survive and pass on their genes. And, a mix of that so that we have certain plants susceptible to one disease but not to another. So you want that mix. And remember, the sexual reproduction is advantageous to an organism only if it happens with someone other than itself. They're going to think, okay, yeah, duh, you can't have sex with yourself and impregnate yourself. Well, that's the deal. Plants actually can. Outbreeding is good, outbreeding is bad, but flowers actually are hermaphrodites. Now, the function of the flower is to attract pollinators with colorful petals and scent and nectar and pollen. So they're very brightly colored. Certain flowers attract different pollinators depending on their color and their shape and their size. But this is kind of the basic anatomy of a flower. And the next slide has a little bit more information about it. But you have in the middle, you've got the stigma, which is the top here. Okay, that's where the pollen lands. Then you have the style, which is this long tube going down into the ovary. If you want to use human terms and you're more familiar with biology, the stigma would be the kind of the entrance into the uterus, and then the style would be the fallopian tube, and then the ovary would obviously be the ovary. So altogether, that's called the pistil or the carpel. Then we have the male part, which has an anther and a filament. Okay, so that's called the stamen altogether. But the filament is just the part that connects it to the flower. And the anther has the pollen actually on it, so that's the sperm. And then the brightly colored bits, the petal and the sepal, kind of encasing the flower. I don't need you to know all the pieces of a flower. I won't show you a picture of a flower and have you identify it all. but you should know which of these is the male and the female. So here's a more high quality picture of all the pieces here, okay? Again, this is a perfect flower. So we call flowers perfect if they have both male and female parts. I know it's a silly term, but that's the idea, is that it's complete in some way, which means that a flower can actually pollinate itself. Most flowers are hermaphrodites. There are flowers that only have male parts or female parts, but the large majority of them have both parts. Again, it's got in the middle, the ovary, the stigma, the style, etc. And it has the petals that will attract the pollinators, and the anther and the filament, which produces sperm. So plants, because of this, have to have a strategy to avoid self-pollination. They don't want to pollinate themselves because pollinating themselves with their own genes does not create any genetic diversity. So they have strategies to avoid self-pollination. One of them is timing. So the idea would be that maybe during the first part of their life, they put out their ovary part, and then the second part of their life, they put out their male parts, which means that the younger flowers will get pollinated by the older flowers in a different plant. Or morphologically, the structure of the male and female organs prevents self-pollination. Maybe it's an imperfect flower, or the stamen are really far away from the ovary, and it would take effort to get that onto it. Another one is biochemical, and we'll talk a little bit more about that, but the idea is that there's a chemical on the surface of the pollen and the stigma or style that prevent pollen tube germination. Basically, they're incompatible with themselves, which is a good thing in terms of wanting to spread your genes around and not just create an inbred population. So I'm going to move to the next slide here. An example of a strategy to avoid self-pollination. So first, here's this picture of a flower. It has the stigma far above the anthers, so it's harder for the pollen to get on top of that. But let's say it does. Usually what happens is that when the pollen lands on there, it'll put this little tube down through the style, and it'll pop up into the ovary and fertilize an egg in the ovary. But if you look here, so it says this S1, S2, okay? So S1, S2 means that those are the genes of the plant with the stigma. And if pollen is coming from S3 or S4, which are two different plants, you're like, oh, great, we can totally accept those. Both of those will be able to pollinate the plant. But if something comes along that has S1 genes, which is the same as this plant, it has a chemical blocker that blocks that pollen tube from growing. If it was S1, S2, meaning it would be the same, maybe the sperm from this plant itself, then both of those pollen tubes are blocked. It basically detects, it has a specific thing that it can detect that prevents the pollen tube from growing. So how do plants get pollen to other individuals? Well, animal pollination is very common. It's where the flowers are large and showy. Flower shapes attract extremely specific kinds of animal pollinators. And bees, flies, and wasps and birds and some mammals will pollinate. A little bit less common is wind pollination. Flowers are small and grouped together. It's not a very efficient method because it's very chancy and wasteful. This has to do with grass and C4 plants like corn. So they would do that. Many plants and pollinators co-evolve together. So coevolution is the interaction between two different species as selective forces on each other, resulting in adaptations that increase interdependency. There are some pollinators that will only pollinate a single species of plant because they've co-evolved over the years, so its specific shape of its beak or something will only work in that flower. Animal flowering and plant interaction is a classic example of coevolution because plants, they evolve these elaborate methods to attract animal pollinators. And then the animals, who also benefit from pollination, evolve specialized body parts and behaviors that aid plant pollination. So why do animals pollinate? Well, they get a reward in exchange for moving their pollen to another flower. That reward is nectar, a sugary solution produced in special flower glands called nectaries. You may have seen hummingbirds, trying to get nectar out of these very deep flowers. Nectar concentration that matches the energy requirements of the pollinator. So bird and bee pollinated flowers have different sugar concentrations than each other. So a bird will maybe have a higher sugar content, while the bees will only have a little bit of sugar. Pollen is also high in protein and is eaten by some pollinators. And this is not great for the plant if they want to spread that pollen. So flowers often produce two kinds of pollen, a normal and one that's fertile, and also a sterile but tasty kind for the insects. The insect eats the tasty one and then spreads the normal one. Pollination is important to agroecology because 72% of crop plants require bee-mediated pollination, and 35% of human foods is dependent on bees for pollination. So if we had no bees, it would reduce our food by over 35%, and we'd miss out on a lot of things we really need, and want, like blueberries and strawberries and tomatoes and other things that really require pollinators. So how do you increase pollinators in agroecosystems? Encouraging things that produce nectar. So, allowing plants that produce nectar to grow near your garden or to grow things in your garden like that. Integrating woody and herbaceous plants for early and late blooming flowers, trying to keep something flowering all year round. Also encouraging nesting habitat for birds, like undisturbed open sandy soil, old trees, snags, or woodlands. And finally, keeping the environment free of toxic insecticides, like adopting the integrated pest management or organic practices we talked about, using biological controls, or selective insecticides. So next we're going to talk about genetics. We talked about that a little bit with pollination, and now we're going to go into in depth with it. But often genetics is described as the hard stuff in agroecology. And so we're going to be talking about the basics of genetics, these single gene, polygene traits, plant breeding, trait selection, genetic modification in the next few weeks. And I know it's a little scary, and I don't want you guys to be scared away because genetics is hard. There are reasons to learn the hard stuff for agroecology. Understanding DNA, mutations, genome sequences, it's an important part of being human in today's world because you really can't get around the fact that genetics are very important to both, our livelihoods and to agroecology in general. All of our food is affected by genetics. Understanding the genetic base of disease is also part of your lives. So genetic modification, whether laboratory or breeding-based, is an intrinsic part of the agricultural progress. So to start, we're going to talk about Gregor Mendel. Gregor Mendel was a monk who pioneered the study of heredity. He crossed garden peas and analyzed their offspring for these heritable traits. He derived these two important concepts: the concept of the segregation of two parental forms of a gene, so that parents were passing on genes, each parent passes on a gene. Also, this independent assortment of two or more parental traits among offspring. So note that Gregor Mendel lived from 1822 to 1884. Mendel's work, unfortunately, was ignored until 1900. Why? Well, he was before his time. No one in that time knew about the cellular equipment that determined heredity. We couldn't see genes or DNA at that point. His experiments would explain simple inheritance, but it was hard for others to understand how his principles could be applied to more complex traits. And he was discouraged by other scientists because he wasn't actually a scientist. He was a monk, but he was getting scientific results. He obtained similar results in some other species, but not in hawkweeds and bees. Now, hawkweeds and bees had he couldn't find any traits that matched up to his patterns he was looking for, and that's because they had a little bit of a different type of heredity. And so he struggled with certain species, but that doesn't mean he was wrong. He just had trouble explaining genetics and everything, which we still do in even in this time. Mendel made two innovations to the science of genetics. He developed pure lines, which means lines that only had specific traits that basically were genetically identical to each other. And he counted his results and he used statistics to analyze his data. We're going to go through the 7 traits on the next slide. So, what Mendel did, now it wasn't called this at this time, but he actually was identifying to phenotype specific traits that were an either or. So, for instance, if you look here, flower color, dominant. Purple is dominant, and white is recessive. We'll get to what dominant and recessive means. But it means that he would see plants that had a purple flower or plants that had a white flower, but he did not see anything that had in between a violet color flower. It was either or. So he was looking for these either or traits, one or the other. Flower position, where was it on the stalk? A yellow or green pea color. Again, it was not both yellow and green. It was either yellow or green, or no yellowish green at all. Round or wrinkled, again, one or the other. The pod color was either green or yellow. The pod shape was inflated or constricted, and the pod height and the plant height was either tall or short. This wasn't like a little bit taller, a little bit shorter. These were like dwarf plants or really super tall plants. And so those were the ones he was looking at. Now, the top ones here are dominant, and the bottom ones are recessive. And what does that mean? Well, we're going to get into that in a minute, but the idea is that when you write out a genotype, now the genes of the plant, you're writing out big P and little p, okay, or big G and little g. You don't have to remember which one means what. The idea is that usually capital letters are used for dominance, and lowercase letters are used for recessives. You usually use the same letter to represent one gene. So a phenotype is what you see. A genotype is what actually goes into the plant. So when you make a plant, it has one copy of the gene. When you breed a plant, one copy of the gene from its mom, that's the egg. One copy of his gene from the dad, and that's the sperm. So hopefully we're all on board with that basic genetic concept. If the gene from the mom and the gene from the dad are both dominant, then you're going to get a purple flower. Okay, the purple is the dominant trait. It's called homozygous. And we'll get to that. But this is one of the pure lines. Okay, so this purple meant that he bred the purple over and over and over until he didn't have any white genes in there at all. So basically, this purple could never create a white. And the white meant that it had both egg and sperm that was recessive from the mom and dad. Okay, so he bred whites over and over and over, and basically now all of them were white and they could not create any purple, which generally the whites can't because they're recessive. So it's a homozygous, the same zygous. And what it puts out is only little p, and what it puts out is only little p, big P, big P. And so you can see the difference there. All right, so he takes these pure lines, and so this is what's called the F1 generation, okay? So you take a pure line, big P, completely purple plant, has all dominant purple genes. Okay, so it has a purple gene from the mom and a purple gene from the dad. Then you take a white plant that has a white gene from the mom and a white gene from the dad, and you cross them together. When you cross them together, the phenotype stays purple. Because again, what happens is that the purple flower gene is dominant over the white flower gene. So even if there's only one purple flower gene, it will produce enough purple that it drowns out the white. So the white is hidden. Recessive is what it's called. Okay, so you breed two of the pure lines, and you get what's called a heterozygote. Okay, so let's look at this again. Again, if you put a purple plus from the mom and purple from the dad, you will get a purple plant, and that is a big P, a homozygous dominant plant. It will only put out big P eggs, big P sperm. When you combine a mom that is purple and a dad that has given a white, it will still come out purple. This is called a heterozygous plant. You can't see it when you look at it. I could not tell the difference between a homozygous and a heterozygous without doing some sort of genetic test. But for this one, it is a carrier of the white gene. It's hidden that is a recessive gene. Okay, so now it can put out either a purple egg gene or a white egg gene, and a purple sperm gene or a white sperm gene. If both the egg and the sperm are have a white gene in them, then it will come out as little p, homozygous, recessive. You cannot have a heterozygous plant that shows a recessive trait, because if it has a dominant trait, it will show the dominant, it will look like the dominant plant. But you have a homozygous recessive. Homo, again, means the same. Hetero means different. You may notice this from homosexual, heterosexuals attracted to the different sex, homo to the same. So it's just the term here. So the same zygote, a different zygote. Zygote being either an egg or a sperm. All right. So then we have, you take this F1, which has big P, little p as its genotype. It's purple looking, but it has a genotype of big P or little p. When it makes babies, there are two kinds, when it makes gametes being eggs or sperm. When it makes eggs or sperm, there are two kinds. They're either going to, make, pollen's either going to make a big P or a little p allele. That's random. Flip of a coin. And then, the eggs will either have the big P or the little p allele. And so this is what's called a Punnett square, this box that you see here. There's two big P's, there's a big P and a little p. These are the things that could be sperm. And then in the pink, you see big P, little p. That's what the egg could be. You combine them together to find the genotype of the babies that come off of this. So after selfing a bunch of these, so basically self-fertilizing, you're trying not to get any other pollen from other plants. He would self them, and then he'd come out with a 3-to-1 ratio. It's not like for every 3 there was 1, but you get something like a 33-to-11 setup, or a 300-to-100 comparison here. And so what you see here is that there's one that's homozygous dominant, two that are heterozygous, and one that is homozygous recessive. And that's because when you combine these two, the big P and the big P of the dominant P's from both plants, you come out with a homozygous dominant. And then there's two ways that you can make a big P, little p, either a mother that has a big mother with a dominant and a father with a recessive, or a father with a dominant and a mother with a recessive. So there's two different ways, and that's why you get two of them. And then there's only one way to make the little p, and that's if both of the little p's come together. The first law: traits are determined by a pair of factors which segregate during gamete formation and are reunited at fertilization. So what does this mean? It means, so these are, he didn't call them that before. He said traits are determined by a pair of factors. These are what we know now as gene alleles. Okay, so a dominant allele will be, is the purple, and a recessive allele is the white. Okay, but again, these different alleles are the reunited. So when it makes the egg and sperm, they separate. Okay, they segregate during gamete formation. So now you have either big P or little p for each egg and sperm, and when they come back together, they will reunite at fertilization when they make the egg and they make the baby, and now you have big P, little p, etc. Okay. Here's an example with Mendel's real data. Take a tall plant, homozygous dominant, and a dwarf plant, homozygous recessive. You cross them together, you get all tall. You cross those again together, and you get 3/4 tall and 1/4 dwarf, a 3-to-1 ratio. And this is shown again by the Punnett square. You see 25% of the time you get this, 50% of the time you get this, and then 75% of the time you get a tall plant, and then 25% of the time you get a dwarf plant. A lot of people thought that at that time that everything was incomplete dominance. So if there's incomplete dominance, then you actually have, when you combine a dominant and a recessive, you actually get an in-between state. So a red and a pink, you get red and a white, you get a pink, and then you would have 1/4 red, 50% pink, and 1/4 white. Now, this is, again, not true for things with complete dominance, because if it was complete dominance, it would be all three of these would be red. All right, so now we're going to go through Mendel's second law, which is a little bit more complicated, but the idea is that this is independent assortment. When two or more characteristics are inherited, the individual hereditary factors assort independently during gamete production. What does that mean? It means if you're looking at a plant that has both smooth seeds, which is a dominant, and yellow seeds, which is a dominant, and you cross it with a plant that has wrinkled seeds, which is a recessive, and green seeds, which is a recessive, you are going to get all smooth yellow to start out with, but now you have both the W, which represents the smooth gene, and G, which represents the green seed, okay? The green or yellow seed. And now you have a larger Punnett square. What happens is that you can either get, so there's 4 different types of gametes that each one can make. So the mom, which is, let's say the mom is on the top here, the mom can make either a dominant-dominant a dominant recessive, a recessive dominant, or two recessives. And then the dad can make sperm that has a dominant recessive, a recessive dominant, or a recessive recessive. Again, the gametes are independently assorting. You don't always get a dominant dominant. You don't always get it's random which gene goes where. So you have 1/4 of each of if you took all the sperm and you typed them, you'd have a 1/4 of them would be homozygous. This would not be homozygous. 1/4 of them would have both dominants. 1/4 of them would have one dominant, one recessive. 1/4 would have one recessive, one dominant, and then 1/4 would have recessive recessive. What you get here, because of this more complex thing, is now you have to combine them all across, okay? So you get 9/16 are going to be the dominant, so 9 out of 16, and they'll be dominant, yellow and smooth. But 3/16 are going to be one recessive and one dominant. 3/16 are going to be the other dominant and then recessive, and then 1/16 is going to be completely recessive. So let's look at a better diagram of this. So this is kind of a better understanding of dihybrid crosses, okay? So here you can see the big R, little r, big Y, little y. Again, it's using different letters for it than the last one, but it doesn't matter that much. It's showing all the gametes that so this is the combination of these two homozygous plants. Homozygous dominant plus a homozygous recessive equals these plants. And now we have these plants that look like the dominant one, but are heterozygotes. And they can produce these. So the mom here can now produce these gametes as eggs, and then the dad here can produce these gametes as sperm. And then if you can combine them, you see that on the outside of the square here where they combine, they all look yellow. But then if you get down into the middle here, you have a yellow, you have a green, but it's smooth. And then, or you have a yellow, but it's wrinkled. And then you only have one that, of the 16 squares there, that is wrinkled and green, the recessive, okay? Well, we'll walk through this more, but here's another one that you can look at. Again, this is just showing, again, the 1/4, 1/4, 1/4, 1/4. And then when you combine them, you see all of the these are the genotypes. Again, you can't see the genotypes when you are crossing them. You're just seeing what they look like. But this is what the genotypes will be. And then you can see why they segregate. So think of it as sort of a math problem here, okay? If you multiply them together. And we'll do the multiplication in class because it's going to be a little bit complicated to do over the internet. But, so now we're going to go through a little bit about why exactly does this happen. The complication here is that we have to understand now, we saw these two different rules, independent segregation and the segregation of traits. But why is this happening? Well, it has to do with chromosomes, okay? So you have to understand the chromosomal basics of crop genetics. The nucleus contains a cell's genetic material, and genetic material is DNA. DNA is packaged into chromosomes, individual chromosomes. During normal cell function, you can't see the individual chromosomes, but during cell division, chromosomes become visible under the microscope. Chromosomes contain the units of heredity. These are genes. Mendel's particles, his factors he talked about, they parallel the behavior of chromosomes. Actually, what happened was that the traits he saw that segregated independently were traits that were on different chromosomes. The ones that he saw in hawkweeds and bees that were so complex and weird, is because they were on the same chromosome and they had a slightly higher chance of sticking together. They didn't independently sort. We're not going to go through that as much, but this is the reason why we see the independent assortment. So we're going to go through meiosis and how that works. Certain chromosomes will segregate with sexuality in certain organisms. These are called sex chromosomes. And the observation of certain characters will segregate with visually identifiable chromosomes. So here's what the human male karyotype, which just means the human male set of chromosomes, looks like. Okay, there are 23 sets of chromosomes, and each of these chromosomes, one of these little legs is from your mom, and one little leg is from your dad. Okay, so they're called homolog pairs. So we see, though, that there's 23 sets of chromosomes, but that each set has its pair. And again, that's because one's from your mom and one's from your dad. So let's go to the next page. Every somatic cell, so of the body cell, in an animal has the same chromosome number, 2N, which means 2 sets of chromosomes. Gametes, eggs or sperm, have half that many chromosomes, 1N. And when gametes fuse, the chromosome number is restored. So basically, you cut it in half. For females, when you create eggs, which you do at the beginning of your life, you create an egg that has just 23 chromosomes, not pairs of chromosomes, 23 individual chromosomes. And males do the same thing with sperm. They come back together, and now they have 46 chromosomes together. And that baby and us have 46 chromosomes. So here is how somatic cells divide. This is called mitosis, just the division of cells. Okay, so let's say you cut yourself and you need to make some new skin cells. So you have so this is just showing 2 chromosomes, but let's imagine this is happening with all 23, okay? So you have 23 from your mom, 23 from your dad. And what they'll do is when they're getting ready to divide it, they replicate for a brief period of time. There's an extra set of chromosomes. Then they line up, they get pulled apart, and they make 2 diploid cells. They create 2 identical cells at the end. But the next one is meiosis. Meiosis is the process that makes gametes, so eggs or sperm. Meiosis reduces chromosomes to 1/2 the number of chromosomes in a diploid cell. They have 2 cell divisions. It's a little bit different. It results in 4 haploid cells. So what will happen is that now you have your mom and your dad have these different sets. They will replicate like they did in mitosis. But then they line up and they find their partner. Okay, they find the dad's, the one from the dad finds the one from the mom. The one from the, then another chromosome from your dad finds the other chromosome from your mom. Okay, so these now line up and they exchange a little bit of information. This is why you get some, this is called recombination. And then they divide, but instead of dividing and pulling these 2 apart to create 2 identical ones, they divide, so now you have a chromosome from your dad and a chromosome from your mom in different cells. And then again, this separation is random. Whether you get the chromosome from your dad or mom in one of the eggs or sperm is random. Okay, so now another one happens, and now you have, again, just 1 copy of each instead of 2 copies of each, like you see in A. Again, if 2 different genes exist in 1 chromosome, there's no independent assortment, and that's why some genes don't work with Mendel's rules, and it's because of this recombination in B. We're not going to go through that too heavily, though. So again, this is something called ploidy. Ploidy is the number of sets of chromosomes in the cell. We already said 2N. Diploid. We are all diploid animals. And then there's also haploid, which is the gametes, so eggs and sperm. Ploidy, again, so gametes have half the number, haploid, and cells may be polyploid if they have greater than 2 sets of chromosomes. In humans, this occasionally happens at the beginning of, say, the division of embryo, but because it's basically mammals can't handle that, the baby will not continue to progress. Okay, so that's would be a spontaneous miscarriage if there happen to be 3 sets, because it doesn't work. But for plants, it does. This is called induced polyploidy. Many plants that we eat are polyploid. Polyploidy can be used to create new plant hybrids, larger, more productive plants. We saw the strawberries before. A lot of the cultivated strawberries are gigantic, are octoploids. They have 8 sets of genes, which is a lot. And compared to the diploids, they are gigantic. Triploid means that it's hard for plants to create fertile seeds because of the way the meiosis works. It doesn't divide properly. So you can actually induce seedless watermelons by making a triploid watermelon. So the way that DNA works to make what we look like, the reason why genotype ends up as phenotype, genes end up as looks, is because of this idea of this genes turn into proteins. Now, it's a little bit more complex than that, but the idea is that there's a gene on DNA. It's read by RNA. RNA goes into over to the into the cell and makes protein. And that protein may be the protein for brown eyes, it may be the protein for skin color, it may be the protein for attached or detached ear lobes. But it's a little bit more complicated. This concept of one gene creates one protein. So one gene creates one type of trait. But it's a bit more, more complicated than that. Genes may be read by RNA differently, and proteins can combine or interact. But the overall concept is that, in general, one gene will create one protein. When we have a dominant trait, that dominant trait is caused by a single gene creating enough protein that it doesn't matter if there's a second gene on a second chromosome to create it. So why do we or plants or other things look the way they do? Well, it's usually a combination of both genetics and environment. So phenotype results from a combination of environmental effects and genetic effects. Sometimes the environmental effects can be very small. Mendel's peas didn't have a lot of variation depending on whether or not they were hot or cold or other things. But sometimes the environmental effects can be so large. And certain human traits associated with food are examples of genetics plus environment equals phenotype. So here's a good example. If we had the same genotype, so same genes, completely identical plants, if you change the pH of a soil in hydrangea, you can change the flower color. And so a strongly acidic soil would make a purple hydrangea, and a weakly acidic soil would create a pink or white hydrangea. So you don't actually have to plant different species. It's just a reaction to the environment. The genes are reacting with the environment, and that's why the phenotype is different. So that can be a little bit confusing. What's genetic, what's not? So how do you tell whether it's a genetic change versus a phenotypic change? Well, the idea is that adaptation is when a stress put on the environment leads a specific trait to be more dominant in a species. This happens over multiple generations. You don't see this happen in between genetically identical individuals. But an acclimation would mean that local environment stress causes a single plant to change its traits based on the environment. So if you plant a genetically identical plant in high elevation, medium elevation, or low elevation, it will be different heights. These three here are all the same plant, okay? See, all the colored plants are all the same. And depending on where you grow them, they're going to grow differently. And this is a reaction to the environment. It's not because they're actually a different plant. And that's because these plants are a little bit complex. So the traits are complex. Initially, scientists did not think that Mendel's laws could explain complex traits, but they can. Two examples of complex traits are traits that are highly influenced by the environment, which we talked about already, and polygenic traits. But those kind of end up blurring together. The bulk of what we see as phenotypic variation within a single species is the result of polygenic traits. Polygenic traits follow the rules of Mendelism, but each one of them is contributing a little bit. So they're controlled by two or more genes with incomplete dominance. Instead of completely dominating, they'll just add a little bit of a different protein to the mix. They're very strongly influenced by the environment, and so multiple gene inheritance. A good example for this is skin tones. Now, many genes with one or more alleles contribute to this trait. 6 to 8 genes will contribute to skin color, and that's often segregated by location in the world. You've got the people with darker skin evolving in Africa, and people with lighter skin in the more northern areas. And a big piece of this is just the reaction to the sun. People with darker skins tend to do better in the lower latitudes, while people with lighter tones did better in the higher latitudes. Each gene has a small additive effect. So if you have these 6 genes that all tone, you will have a very dark toned skin. If you have these 6 genes that are very light in tone, then you will have a very light skin. And there's a lot of in-betweens. And again, obviously, this is affected by the environment. I'm assuming that a lot of you guys can tan or do change color in the sun. I don't exactly. I have a little bit of an issue. I'm very freckly. Freckles are actually a problem with the creation of melanin in your skin. So you get speckled instead of producing a nice even tan. But this means that you can be very pale but then tan easily, or you can be very dark but not tan as well. And so there's a whole spectrum of how the environment affects things. Another one is human eye color. Now, a lot of people think, but wait, it's just brown and blue, but that's not true. People have a lot of different eye colors and patterns. At least 15 genes contribute to eye color, and actually, I was curious about my daughter, what color her eyes would be, because both my husband and I have hazel eyes. She has brown eyes, but apparently hazel is pretty complicated, and it's hard to predict whether or not it's going to pass on to the next person because there's too many genes affecting it. Polygenic traits tend to be continuous, and what this means is that instead of having either really big or really small, you have a whole range of big to small. Similarly with people's skin tones, you don't have one skin tone, you have a whole range of skin tones that goes from dark to light. Continuous is what it means. It's not either or. So the diversity in apples, for example, is polygenic. You have multiple traits that control the color on the skin. And so that's why you have so many different varieties. So again, polygenic traits are influenced by the environment. This is kind of the classic view of quantitative genetics. Many genes will contribute to a trait, but each gene has a small effect, and the environment is blurring the discrete effects of the individual genes. So because there's multiple genes and the environment's affecting each one individually, then this results in a very complex phenotype that's hard to predict exactly. So next lecture, we're going to be talking about improving plants through breeding, cultivation, hybridization and grafting, marker-assisted breeding, and a variety of other things. And I'm actually going to post a example that you guys are going to be going through before next lecture. Basically, you're going to be looking up your own family tree and heredity, and you're going to be looking at the your own background and seeing how genetics is affecting your family's health and presenting that to class. So that's going to be posted a little bit later. I hope you guys all have a great spring break, and I will see you next Wednesday.